So what I still see a lot is people expecting a genomic code to be able to predict things very clearly. And in my clinical work, I come across that a lot, where I have to spend a lot of time telling people, well, actually, this test is not going to tell you as much as you hope it will. There's a lot of discussion at the moment about newborn screening, and people expect that we'll be able to shed a light on their newborn baby as to what illnesses it will get in the future, and it's a sort of blueprint. That our knowledge about genomic medicine is very biased. So it's based on largely data collections and biobanks that are overwhelmingly Eurocentric. I think there's two problems. One is that the initial population, the initial databases that we set up were very Eurocentric, and because they're now the oldest, they've got the biggest amount of data, and they're often the ones that are able to release that data relatively easily. And so researchers around the world turn to that database, and that perpetuates the cycle a little bit. But I think the other big reason, of course, is that there is a legacy of historical injustices to underserved communities that therefore don't trust genomic medicine in quite the same way as people in the UK might, and therefore are much more sceptical of why should they take part in these sorts of research ventures, meaning that, again, we don't collect that data. So I think we need much more co-production, looking at engaging with communities about what their issues are, what they would want from a venture. And that's not easy to do. It's much easier to send out a kit and try and collect a sample without really going to have to do all the engagement with populations elsewhere to try and diversify your collection. More than 80% of all genomic studies and more than 95% of a particular study called a GWAS study, or that looks at particular markers in the genetic code, are around the world are done on people of Northern European ancestry or European ancestry, whereas they actually form a minority population in the whole world. So I'm looking at why it's so difficult to make our genomic collections, upon which we base genomic medicine, more diverse and appropriate to populations other than myself of European ancestry. We know that ethnicity is a complex social construct that's much more to do with your geography and your religion, perhaps, and your community than your genetic ancestry. Yet, forms that we use regularly in daily practice and that are mandated within the NHS to be used often ask about skin colour, geography, and nationality as a means to try and assess whether enough diversity has been reached in a particular sample, when those questions should also be asking about socio-demographic factors, really, because I think everyone can see when you say it like that, that skin colour can't be the only measure of diversity.